Hi everyone, and welcome to our video on gene therapy. This is part of our Module 6 Part 3 series looking at manipulating genomes, and in this video we have a look at specification reference 6.1.3H, the principles of and potential for gene therapy in medicine. Before we delve into anything to do with processes, let's understand what on earth we mean by this phrase gene therapy. And quite simply, this is altering a person's genetic material through a method. Often this is being used to treat a disease. So basically in gene therapy, what we're going to be doing is changing someone's genetic material. In terms of how we do this, then quite frequently what we're going to be doing is actually identifying a condition where it's caused by the presence of a faulty allele of a particular gene. And what we're going to do as a treatment for that is insert the functional allele into the cells that are affected. Now, when we've inserted that functional allele into those cells, then the gene will be expressed, and that will lead to the elimination of the symptoms of that condition. A different way that we could approach this is by using something called interference RNA. And the whole idea of this is that it's going to actually silence certain genes by blocking their translation. And a good example of where that's actually been employed in the past is in AIDS patients with cytomegalovirus infections. So what we were able to do is use this interference RNA to actually block the CMV replication by just basically stopping it from being translated. When it comes to ways to approach gene therapy, we've got two ways that you need to know about: somatic cell gene therapy and then the germline gene therapy. So let's start off with the somatic cell gene therapy first of all. Now, hopefully we remember that where we're talking somatic cells, those are basically the normal body cells. If we think about one of the key examples here, let's go with cystic fibrosis. Now, cystic fibrosis is a disorder caused by inheriting two faulty alleles which are recessive. So in order to actually have cystic fibrosis, you need both of those faulty recessive alleles to be present. Now, what it means if you do have both of those recessive cystic fibrosis alleles is that you're not going to be able to produce the protein CFTR. And this is a transport protein that we find within the membranes of our cells. So anyone with cystic fibrosis lacks the actual protein CFTR. And one of the approaches to tackling cystic fibrosis is to use this somatic cell gene therapy. And what we're going to do there is we're actually going to insert these functioning alleles into the body cells. Now, the key thing to remember here is that when we're talking somatic cell gene therapy, whatever we do is not going to be passed on to the offspring. We're not changing the genome permanently. We are just changing the alleles present within certain cells. So it won't be passed on to any offspring, but it can help treat these conditions. So as far as the cystic fibrosis goes, then generally we use like an inhaler, which is then going to deliver these functional alleles to the cells lining the respiratory system. And if we know anything about cystic fibrosis from lower down the school, hopefully we remember that this is going to cause this thick, sticky mucus because the transport of the ions and the water across those membranes isn't functioning correctly because of the lack of that CFTR protein. So what we find here is that by delivering these functioning alleles to the cells lining the respiratory system, they're then going to be expressing the CFTR gene, and that means that we're going to have that protein inserted into the membranes, and we're going to then at least partially resolve the issue of that thick, sticky mucus. The way that we're actually going to make sure these functional alleles reach their intended destination is through the use of a liposome. Now, if we look at liposome, then hopefully we're thinking lip stands like lipids, and it does, because it involves a lipid structure. So liposomes are going to package those functional CFTR alleles, and then the inhaler delivers them to the respiratory system. The idea being that some of those liposomes are going to pass through the plasma membranes of those cells lining our respiratory tract, and if they continue to pass through the nuclear envelope, that's where we can insert the CFTR allele into the host's genome. As soon as we've got it inserted, that can then be expressed, and we will then produce that CFTR protein, which can then be implanted into the plasma membrane of those cells. There is a big problem with this treatment though, and the first one is that the lining of your respiratory tract isn't overly stable. Because of the wear and tear it gets from this constant movement of obviously gases through it, etc., then we end up replacing those cells every 10 to 14 days. So what that means is any of those cells that had gained the functional allele then will need to be replaced, because every 10 to 14 days those ones with the functioning allele are going to be obviously removed by the body and replaced with ones using our original DNA, and therefore won't have the functioning allele present again. So basically any of these treatments for our somatic cell gene therapy need this repeated exposure to that functional allele. And obviously depending on which particular aspect of the body we're talking about will determine how regularly these treatments will need repeating. The other option for a delivery aside from our liposomes is using a virus delivery system. So we can use these viruses as a vector for delivering that allele. The problem of using a virus is the fact that obviously we are introducing a viral particle to our body. Now, our immune system may still recognise the fact that this is a virus, it is something not belonging to self, and as a result of that, we may mount an immune response to it. And obviously, if we're then exposing our body to this particular virus delivery system over and over again, what we may well end up doing is developing an immunity to the vector, which means those future deliveries using that same virus delivery method just can't take place. Because if our immune system is too effective at fighting off this virus, then we just won't be able to insert the functional allele. The next issue is that when the virus is actually inserting the allele into the genome, it might be put in the wrong place. So if we ended up with the actual allele we're trying to introduce in a regulatory region, that might lead to uncontrollable cell division and therefore increased risk of cancer. It might be inserted into the regulatory region for another gene and therefore disrupt another gene being expressed, which could have unintended consequences. We could also potentially see this frame shift leading to altered triplets. So if we've got a frame shift occurring, then we're going to see altered expression of other proteins, or it could switch on or switch off different genes nearby. None of these are particularly ideal scenarios. What they're currently looking at as a strand of research at the moment is developing something they're terming artificial chromosomes, or HAC. And these artificial chromosomes are going to act as a delivery system for these functional alleles. And the reason that we're looking at developing these is that we would then avoid those problems of the virus vector and its insertion, and has another bonus that we could actually use very large or complex genes, or if there's a condition that's obviously regulated by more than one gene, so many other genes, we could actually insert a whole range of them together on this artificial chromosome that we have created in the lab. It's still relatively early days on this one, so watch this space and we shall see what happens. The second option is a germline gene therapy. Now, germline gene therapy is all about inserting the functional alleles into either the gamete or the zygote. So we're going right back to the very beginning of the organism's existence at this point. Now, when we alter the actual genome of the gamete or the zygote, this is a permanent change. And every cell that then comes from that altered one is going to have the altered allele present. So what we've got to bear in mind is we're not just changing one cell, we're going to be changing that cell, every cell that then comes after it, and any cells of future offspring of that particular organism as well. So this has very wide-reaching effects. And because of this, it's illegal in humans. So we are not allowed to carry out germline gene therapy on humans because it is a permanent change that impacts the future people who've not given consent. If you are a zygote, you cannot give consent. You are a fertilized cell to all intents and purposes. So quite clearly, we don't have consent to be able to carry out something that is this long-standing. Now, the other concern that we have as far as the germline gene therapy goes is linked into that gene insertion, potentially increasing risk of cancer or expression of other genes, as we've already mentioned. So one of the best things I suggest you do for this little section of gene therapy is to actually learn this little table. So all I've done here is I've just summarised 4 key facts about the somatic and the germline gene therapies, just so that we've got that direct comparison between them. So somatic, which you remember are the regular body cells, these are going to be the ones that can't be inherited. We will modify it in the individual who is receiving the treatment, but it will not be inherited by any offspring. Whereas the germline, which is obviously the zygote or the gametes, then that one is going to be passed on. So that will be inherited by future offspring. Obviously, somatic is inserting into the body cells, whereas germline into the zygote or the gamete. Somatic, only some cells will get that allele, whereas in our germline, because we're altering that original zygote or gamete, all cells get that functional allele, and therefore germline is a long-term, whereas the somatic is only a very short-term treatment. So make sure that you do understand the difference between these, where we'd use them and their potential implications. And if you do get a question on any of the A-level papers asking you to compare these, so you have basically outlined this table asking you to give 2 differences between them, please make sure that you're writing it so that the differences are lined up on one of these horizontal rows. If you were to write short term and then next to it in the germline column put can be inherited, you're not getting the marks because that's not actually a comparison. So whatever you're comparing on that paper, you've got to put in those horizontal rows so it reads as a comparison on that row. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next one is uploaded, and then head on over to the website where you can find a range of other resources to help you in your A-level biology studies.